So now you have heard me talk about various applications of trees, but of course it's a whole different thing to hear a scientist who actually uses graphs and trees in their current research. And that is why I'm very happy today to welcome Gulnara Tagirdzhanova from the Department of Biological Sciences at the U of A. She does research on the evolution of symbiosis, and she will tell us about how graph theory is used in biology and how she uses graphs and, in particular, trees in her current research.
Okay, hello everyone. I'm here to give you a quick introduction in how graphs and graph theory is used in biology. I was planning to cover two topics. First is graphs in visualization and sort of presenting biological knowledge, and second, graphs and data analysis in biology. So it just so happens that directed graphs are the default way of presenting biological knowledge. Why is it the case? Well, we have lots of different objects and they are interacting among each other in different ways. So, when we are trying to present all of these different networks, these objects become nodes and the interaction become edges. This is the case for biological systems in different levels of organization, starting on macro level with interactions between organisms. So this is an example of a food web, something that you might remember from high school biology textbook. And here you show this trophic interactions or interactions of eating each other between different species within an ecosystem. But the same principle also applies to microscopic level. So here, for example, you see a sort of map of protein signaling pathways. Here, all the vertices are different proteins, and the edges are the way how these proteins interact with each other in a process of transmitting signal within a biological cell. I just wanted to give you two more examples of graphs in biology, the ones that I work with the most. The first are metabolic maps. So here on the slide you see a metabolic map of a bacterial cell. It's super complicated. It describes all different molecules that are involved in this cell, and this is not really easy to work with, so I just wanted to zoom in into a more specific part of this metabolic map. So this represents a metabolic pathways around riboflavin, a B6 vitamin. And here you see another directed graph where every vertex represents a molecule and every arrow or every edge is a chemical reaction. So you see how within a cell different molecules are transformed one into another with the aid of some proteins. So this is example number one. Example number two are phylogenetic trees. Phylogenetic trees are arguably the most common type of graphs that you see in biology, and they are diagrams showing evolutionary relationships among different species. So here you see the leaves. They represent extant species, the species that are currently living and the ones that we can collect our data on. Then you have branches. The branches are edges that represent extinct lineages. So all of the species and that were present in previous times. And the root is a common ancestor of this extant species we are working with. So generally the assumption of a phylogenetic tree is that on x-axis you have time, evolutionary time, and the species are evolving and changing along this axis to arrive from the ancestors, from the root, to the leaves, to the species that we are currently working on. So this tree that you see on the slide is the one from the paper I'm currently working on. And this is the most complete, up-to-date tree of life meaning that it includes all biological life that we are aware of. Here I just indicated with an arrow the part of a tree that encompasses all animals, and everything else is just a vast diversity of bacteria and whatnot. Okay, so that was the first part about graphs used as a way of graphically representing biological data. The second part is more about how we actually derive on the point of having that data. There are multiple examples of how graphs and network analysis is used in biology. I just wanted to focus on one specific example that I am familiar with, and this is a usage of graph theory in bioinformatics. So, to introduce a problem, we have a chromosome. A chromosome is a chain of millions and millions of nucleotides that are arranged in that linear fashion. And as you probably remember, there are 4 types of nucleotides: A, C, Gs, and Ts. We want to record the exact sequence of these nucleotides on the chromosome, and we can do that, but we only can do this sequencing procedure with short pieces of DNA, about 200 nucleotide long. So what do? We take lots of copies of the chromosome, we break each of them in short 200 nucleotide pieces, and we sequence them. But now we have all of these millions of reads, and we need to reconstruct the original long sequence of a chromosome. This problem is called genome assembly, and the way how this problem is currently solved is through application of de Bruijn graphs. De Bruijn graph is a directed graph representing overlaps between sequences of symbols. So how does it work? We break all sequences into k-mers, and these k-mers becomes vertices. So on this picture that I have in the slide, the k equals 4. So after that, we add edges between all nodes that overlap by k minus 1. And that creates this graph. So the graph on the picture is for this short sequence of nucleotides. And as you see, there is a cycle within that graph, and that cycle is caused by the fact that there is a sequence TAGAC that is repeated twice. So after we created this graph, the goal is to simplify it as much as possible, because the data presented in a graph form is not really easy to use for any downstream application. So to simplify it, we try to find an Eulerian path into this graph. So we will arrive in a collection of long sequences that we can then use for any sort of genomic analysis we're interested in. So on the slide, you see sort of like a toy example. This is how a de Bruijn graph looks in a more real-life example. This is the genome of Escherichia coli, probably the most well-studied bacteria. And you see that there are some long colored stretches on the graph. These are vertices and the edges that connect them, and then there are some loops and whatnot. But this is how in general the Bruijn graph works in bioinformatics. Okay, so that was a few application of graph theory in biology. There are many, many more. And thank you for your attention. And if you want to know more about bioinformatics, feel free to contact me by my email. Thank you.